Thanks, ma'am, for the introduction, and thanks the organizers for inviting me. Uh, Before I start, I must acknowledge my today's talk is very special because first time I have a special audience in the audience who is listening to my talk for the first time, my daughter. So she will judge me for sure, and I promised her that I will try to entertain her as much as possible and not to bore her. So hopefully, I know it's COVID, it's disease, but let's also have some fun. So I will divide last maybe 5 6 years into three groups: BCC and AC. So this is BC, when everybody was having fun, going to their normal life, but something somewhere was going on. I'm not going to put any theory out there. Something somewhere is going on. Okay. Then one fine day in March, suddenly something happened. Somebody came onto television and says, Corona, started the era of lockdowns, and officially in Indians understood Corona has come, something has come. So now we are very much cautious, washing vegetables with detergents, taking the spray everywhere to kill the virus everywhere and all that. And we are very scared Corona will come inside and kill us, like that. Okay. Then one fine day, something happened called vaccination, and we defeated Corona, and we threw our masks. Mask is gone, Corona is gone, everything is gone. Okay. So this how just shows how ignorant we are about a disease which is we still very little understand about. So right now there are lots of things we know about affecting our brain as well as heart. I'll come to that more about later, but we don't care. There is fire all around when we are gone back to the dancing mode. So this is the ignorance that I'm going to talk about, and right now we are here. We don't even want to know anything about COVID. So when I was continuing my study, my ethical committee actually asked me, ethics actually asked me, why are you continuing? There is no COVID. So why do you need to continue this study? So the question is, are you actually even aware that COVID is actually not only a respiratory disorder? It can affect every and each and every part of your body. I'm not going to read that slide. Probably some of you already know. Dr. Moitra is laughing. Maybe she's smiling. He may already know about it. So I'm not going to go about that. So let's start. So this is for last year. So to convince, basically this study was done just to convince our ethics committee that COVID is important, and mostly my talk will about long COVID or past. But we did one Mendelian randomization study. I'm not going to go detailed about Mendelian randomization study, but we collected the largest possible COVID-19 severity data and data from different disorders, starting from pneumonia to Alzheimer's, Parkinson's, headache, heart failure, all these things from IU Open GWAS, and we showed that there is a strong connection, especially between pneumonia, heart failure, Parkinson's, and chronic fatigue. So if you see this, these blobs without much error bar, that just shows a strong connection of these four. This is pneumonia, this is Parkinson's, this is heart failure, and this is chronic fatigue. These are the characteristics that has direct connection association with COVID. And those who are aware of Mendelian randomization may appreciate this, but those who are not, I can answer that later during lunchtime or somewhere. So on the basis of that, actually we started this study before we did randomization study, but mainly we wanted to focus more on long COVID or PASC, but because our initial ethical clearance were for severe COVID, but as we progressed, we understood COVID is less of a respiratory disease and more of about other organs. So we started more focusing on the long COVID. So sadly, as you can see, the number of participants we could genotype is only 60. Our target was 200 at least. We requested every single person to volunteer, but everybody was asking the same question: COVID is gone, why would I give my sample? So we had a severe problem to sample collection, but our good luck is we could collaborate with GBRC, Gujarat Department of Biotechnology, and they had more than 500 people's data. So we could merge them. And also the problem with the PASC cohort was, right now there is no available PASC genome or post-COVID complexity based GWAS data available anywhere in the world, let alone India. So we could not get any data. So for that we have to use population control. and we use Genome Asia data. I'll come to that in a minute. So before talking about the GWAS results, I would like to mention, yes, Dr. Mohita was mentioning something in the morning, and I completely agree with that. The current array that are available are by the waste, for the waste, to the waste. So the thing that we can capture, it is limited, but whatever we can capture, that results is based on that. So we divided our study into two cohorts. Sorry, slide is not moving. The CVIT cohort as well as the PASS cohort, I don't know why the Manhattan plot is not loading. Now it's there. Okay, so it's the CVIT cohort where we compare the asymptomatic versus mild to moderate versus severe. And this is these are the different chromosomes, and this is the minus log P. And I have only pointed out those which are above minus log P 8, the 7 boundary, this red line, and many of them actually has connection with neurological, like Alzheimer's, and then memory function and all that. This is for the CVIT cohort. Now for the CVIT cohort, when we perform the enrichment analysis, we found there are several things that are being enriched, like long-term depression, circadian entrainment, glutaminergic synapse, cholinergic synapse, and all that. And also, we found cardiomyopathy. So if you look at the geo terms, it came the neurogenesis, nervous system development, generation of neurons. So something has connection with the brain. This is for the CVIT cohort. Before doing the PASS cohort, as I told you, our issue was we had to limit our study to South Asians only. And since we take the data from the Genome Asia, we performed one PCA and restricted our study to a particular group which data matched with our PASS patients. And now when we did the GWAS, as you can see, we had very highly significant some recently we have found. For example, this one EYS, then this again encephalopathy, and if you can see there is a peak here which connects with encephalopathy and OCD and all these things we found out. Overall, there are several cake pathways that are associated with PASS cohort, associated with the neural genesis and cardiomyopathy, so brain and heart. And if you look at the geo terms, the nervous system development, it comes most prominently. So these are the neuropsychiatric disorder pathways that I'm showing. I'm not going to go into details of it. These are the genes that we identified. If you look at the cardiovascular, more genes you can see for cardiovascular disorders. One more interesting thing that we have found, actually that we have shown with this Dr. Priyanka, we have done this for we have done two GWASs before this, which were on European cohort. This ECM interaction, ECM receptor interaction, we have found in European cohorts as well, and we have identified several genes in the PASS cohort that we identified previously for European cohorts as well. So ECM is another factor that we identified. Then if you talk about the pathway analysis, then in all pathways that you can see, this is the neuropsychotic pathways. These are the pathways that we have identified. And another one interesting pathway that we identified is GPCR signaling pathway. So if you remove those neurological pathways out, the second pathway gets prominence is GPCR. So we have several GPCR pathways that we have identified. And another interesting thing that I'm not 100% sure about, but it is coming up. There is a connection between severity of COVID and PASS with telomere length. Now I don't know which direction it is. Individuals who have shorter telomere have more chance, or who has severe COVID has shorter telomere. It can go by the direction. I do not know which direction it goes to. And these data I cross-checked with UK Biobank data, and we have seen there's a significant difference in the individuals who have severe COVID versus who has mild COVID. The telomere length varies. Actually, the individuals who have COVID has a shorter telomere length. Now the question was, the entire data that I showed, everybody can see this is only computational, predictive and all that. So I was looking for a collaborator. I'm computational biology did not do any experimental work. So I was looking for somebody who we can collaborate with and get some transcriptomic data. Obviously, transcriptomic data was there for the blood as well as the, I think there was some blood as well as kidney, some of the data was there for the humans, but we wanted to see with the more experimental. So I collaborated with Dr. Byron Reddy, University of Nebraska Medical Center, and these are the transcriptomes of infected hamsters who are infected with COVID. And then, so this is the result. So for lung, you can see there is a distinct difference between case and control, who are infected versus non-infected. Not so much in the brain. This is for the hamster, not for the humans. And if you look at the enrichment analysis, we did not find any enrichment in the hamster brain transcriptome, but we found lung transcriptome has a lot of enrichment again in the nervous system. So the first question came to my mind, was the tissue, I mean, either ka udhar ho gaya, something. So I asked him, he said he will cross check, and he cross checked, no, tissue is fine. So in the lung tissue transcriptome, we have found a lot of neuronal aspect, but brain we did not find anything. Now next thing I wanted to see is probably many of you are aware about polygenic risk score. We generate a new algorithm for polygenic risk score calculation, APRISE, and we wanted to see whether we can predict genetic propensity for PASC, who can have post-COVID. So for us, we only consider the individuals who are in the first quartile or hello, or the first quartile or 25. Hello, hello. Hello, hello. I think I can. Can you hear me? My voice, I think, is loud enough. At least 3. Huh? Okay. Hello. 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 So we consider the scores which are in the first quartile or 25 percentile as the low risk area, and any score which is above 25 percentile here to be in the high risk area. As the score increases, the risk increases. So if you compare the first with the second quartile, it is significant, and so on. Actually, we showed individuals who has PASC. This is the PASC distribution. This is the no PASC distribution. So you can see PASC, obviously the median is higher than the no PASC, and no PASC individual score is much more narrower than the PASC. Now, so the PASC, some score will actually lower than the non-PASC, but it has a larger distribution. Then we looked at the individual specific variation for these scores, and we found actually among the 60 people, there are 3 people like over here. They had distinctly higher score than the rest, these 3, and among them actually has de novo diabetes, and 2 persons who has another person has diabetes, he has gained weight as well as has diabetes, and the third person has developed newly like anger issues and some breathlessness. So the scores match, and again breathlessness is coming. It matches with our chronic tiredness, that thing. This is what I think is very interesting, that we took the SNPs that we identified in PASC versus no PASC GWAS and applied that for genome Asia data. So we used only those SNPs which are p-value less than 10 to the power minus 8, and we see how the polygenic score varies across the world for those SNPs. So if you look at this is British people, Pathan, and all the caste population in India. Then comes the tribal groups in India, then Northeast India, then Andaman and Nicobaris. These are East Asians. So as you can see, there is significant variation in the propensity for PASC if you go from the caste population to tribal group to Northeast. So and the scores of Northeast population is very similar to the East Asians. One more interesting I would like to highlight. If you look at here, this is an African population who falls in the median. This shows African has the highest diversity, so maybe their scores have a lot of variation, and the variation is somewhere in the middle. And I don't know, maybe some of you are aware, the UK is actually facing a lot of issues from past. Just to give you a context, that past is a problem, and last year, 2023, there were people who was protesting against not to stop research on COVID. You should continue researching on COVID. We know very less about it till now. And I'll show you a couple of slides. So this is the interactome analysis that currently my student Shilashmi is doing. So in here, we looked at the interaction of the virus and the human proteome year by year. So if you look at the alpha variant in 2020, and the enrichment has nothing to do with brain or heart. When you move from alpha to delta and omicron, now it shows your heart and brain coming more and more. So the interaction has changed. Interaction has evolved over time. And if you look at the last variant we know about, the Omicron variant, which is current variant as well, and so extracellular matrix protein and the brain and heart is more prominent in the Omicron. So we thought Omicron is our savior. Actually, it may not be the case. Omicron is causing more problem. Omicron is not causing problem directly or on your face. It is causing problem in the long-term way. That's what this analysis shows. Now what? What to do next? So we are going to make an AI/ML based model of the human interactomes. Also, we have a plan right now in ethical way. The funding is approved already by our university. So we are going to look at actual patients, Parkinson's patients, and in our hospital, and see whether they had any history of COVID-19. Whether the Parkinson's patients with COVID have certain genetic signature versus Parkinson's patients without COVID they don't have. Similar thing we will do for some pulmonary disorder, respiratory state disorder, and this is with the collaboration with KMC Manipal. What we will look at is same aspect. We will look at the patients with and without history of ARDS, whether they had COVID versus they don't have COVID, and try to find any signature. Yeah, my slide is up. So this slide is about. So let's stop ignoring the past in the room. We have ignored it a lot. Thanks. Any question? Yes.
Thank you very much. It's really an issue. So I personally, born with a particular syndrome, Julian Bayer syndrome, and which is really associated with the COVID-19. Do you have any kind of information about it? Julian Bayer syndrome.
I can check, but I don't right now.
Some papers that come up with associated with COVID-19.
It's possible.
Nervous system and this syndrome. I didn't listen it earlier before.
But syndrome means it's not monogenic, right? Because this is not a monogenic condition.
Yeah. That's what I asked.
Yeah.
If you have any kind of
because obviously COVID is a multifactorial condition. It's not a monogenic condition. So that's why we have to be careful when we interpret. Okay. Thanks.
Our next speaker is Dr. Renuka Subrathala, and he is present assistant professor at the American School of Biotechnology.